Okay, sorry, my computer's being slow. Can you guys hear me?
Yeah.
Awesome. Any questions? Okay. So if there's no questions, I'm just gonna carry on from where we were talking about before. Okay. Oh my goodness. Okay, so we covered the endomembrane system. Just remember that it consists of the nucleus, the endoplasmic reticulum, and we have both the smooth and the rough. Remember, the rough makes proteins. The smooth has all kinds of different functions, including synthesizing lipids, breaking down toxins. And then these things are shipped around with vesicles, which also have a phospholipid bilayer, same as the cell membrane. The Golgi, remember that, oh, in the ER, the rough ER can modify things by adding sugars to make it a glycoprotein or lipids to make it a lipoprotein. And the Golgi can do the same thing. And then the Golgi is basically modifying these proteins and then shipping them off to where they go. So maybe the protein needs to go back to the nucleus to copy the DNA, so it gets shipped there. Maybe it's getting shipped outside of the cell, like components of your saliva or whatever. All right, so this is not part of the endomembrane system, and these things have their own. They generally have their own DNA. All right, come on. All right, so the first thing we're going to talk about is the mitochondria. So mitochondria are organelles. They're where we do cellular respiration, and we're going to cover this in chapter 9 extensively. So all of chapter 9 is nothing but it's called cellular respiration. Respiration is the reason that you breathe. So you take in oxygen, you combine that with the sugars you eat or proteins or whatever, and there's just a different pathway, and we'll go over that later. And that creates ATP and some byproducts like carbon dioxide. So the reason that you breathe oxygen and exhale carbon dioxide and eat food is to make this beautiful molecule called adenosine triphosphate, or ATP. This is your energy currency. And most of you guys got that right on the protist question lab. Okay, so this is where oxygen comes in, and we make ATP. Mitochondria are only found in eukaryotes. So prokaryotes still do this, but they use their cell wall. Remember, all prokaryotes have a cell wall and their cell membrane. To act like a mitochondria. In fact, a lot of scientists believe that mitochondria are actually ancient prokaryotes, ancient bacteria. There's some reasons for that. Well, they don't have a cell wall. They've lost that through evolution, but they do have DNA. So they have their own DNA. That DNA is not enough to allow them to live, but it is enough to allow them to make ATP. So, you know, it's kind of odd that it has its own DNA where other organelles don't. And I used mitochondrial DNA to get my master's degree. I did a study on turtles, and then I also used it to get my PhD, where not only did I look at mitochondria, but I looked at the DNA in the nucleus of all fish, all bony fish. So I did a comparison of all the DNA of all of bony fish in the world, and then constructed how they were related. But mitochondrial DNA can use to figure out, who the baby's daddy is and stuff like that. And in humans, mitochondrial DNA is only transferred from the mother. So every one of you guys that are listening to me right now, you got your mitochondrial DNA only from your mother. And the reason is because in humans, sperms are really small and the egg is really big. And remember, we talked about the cell theory that all cells come from other cells. Well, all organelles have to come from other organelles as well. And so your mother packages the mitochondria, and whenever the egg is fertilized and becomes a zygote, and that's where you started, and then you divided through a process called mitosis, and we'll cover this in chapter 2 and 12. All of the mitochondria that were passed on to all of the daughter cells from when you were conceived, even if you were conceived in a test tube, it works the same way. All those mitochondria came from your mother. It's not true for all animals. Some animals, like fruit flies, the sperm is bigger, and so it carries the organelles. But in humans, this is how it works. So not only do they have their own DNA, but in humans, all that DNA comes from the mother. And we can trace this back in time by comparing all females on Earth from different races. And we can, there's actually a study, it's called Mitochondrial Eve. And it traced back the ancestor of humans to Africa around 200,000 years ago. So this is where humans, the current human population originated. That doesn't mean that there weren't humans before. There could have been a whole bunch of humans, and then maybe a disease came along, you know, like a big one, and killed off everyone that wasn't resistant to it. And then they started a new population and recolonized the earth from that small population. So that's possible. We don't know what happened before that. We just know what the genetics say. All right, so mitochondria have two membranes, an outer membrane and an inner membrane, and that's really important. So you're going to learn this in chapter 9, but what the job of mitochondria, I'm just going to turn this into a mitochondria. So this is now a mitochondria. It's no longer a fertilized egg. We have the outer membrane here, and then the inner membrane here. And so what happens is that just like your stomach can pump protons into it to make it more acidic, the mitochondria actually pumps protons across this inner membrane, and that builds up a positive charge on one side and a negative charge on the other side. And so it's this difference in charge that drives, there's tiny motors, believe it or not, just like the motor that you'd find in your electric car or your vacuum cleaner. These motors spin just like that, and they generate force to produce ATP. And we'll cover that in detail in chapter 9. But if we go back to bacteria, they do the same thing. They're pumping protons across their membrane, and their cell wall is containing those. So it gets this positive charge and negative charge across it. And they also make ATP this way. So when you take an antibiotic, a lot of times that antibiotic will destroy the cell wall. And without that cell wall, you can't build up that charge. So this charge just dissipates. And because there's no charge buildup, it can't drive the motors. And these motors are the same in bacteria as they are in humans. You could take one out of a bacteria and put it in a human and wouldn't know the difference in a mitochondria. And if there's no buildup of charge, the motors can't spin. It'd be like cutting your electrical cord to your vacuum. And because it can't spin, it can't make ATP. And if you can't make ATP, then you die. And it doesn't take long. If you guys wanted to stop making ATP, all you have to do is hold your breath and you'll stop making ATP because you can't make it without, we can't make much without oxygen. We'll talk about a special case called fermentation, but for the most part, you need a ton of ATP. If you hold your breath, most of your ATP is gone. And, maybe 7, maybe some of you 10 minutes tops, you could go without making ATP. But other than that, you would die. And so that's what happens to bacteria when you take antibiotics. It attacks the cell wall and breaks that down and stops ATP production. The reason it doesn't affect you is why? What's different between you and bacteria?
You don't have a cell wall.
Yeah, you don't have a cell wall. And so because you don't have a cell wall, who cares if it affects your cell wall? Because it would never affect you. So that's how antibiotics work. Some of them, there's other ones that work in protein synthesis and stuff like that, but we'll get to that when we get to chapter 17. All right, so that's why mitochondria have to have an outer and an inner membrane. If it lost one of these membranes, it wouldn't be able to make that charge and it wouldn't be able to drive those motors to make ATP. So these are really important, and we'll get into detail on these when we get to chapter 9. We'll break this whole thing down. But just like the endoplasmic reticulum, it has these folds to increase surface area in it. The name of these, remember those were cisternae. These are called cristae. And then we have the inner membrane and the outer membrane. This is an electron micrograph. And what kind of microscope do you think took this? Scanning or transmitting?
Transmitting?
Yeah, because we can see the inside of it. Otherwise, we wouldn't be able to see the inside of the mitochondria. So this is a cross section. So that's exactly right. Transmitting electron microscope took this. So if I threw this picture on the test, you guys would get that right. And then in between them, we call it the intermembrane space. So we have the outer membrane, the inner membrane, and then the space in between where these plus charges are is called the intermembrane space. So you're going to need to know these locations because they're going to be important in ATP production, especially when we get to chapter 9. So in this chapter, you might as well go ahead and learn the inner and outer membrane and then the intermembrane space. Because let's face it, without ATP, life wouldn't exist. So it's kind of important when you're studying biology, which is the study of life. Okay, so that covers the mitochondria, the chloroplasts. So some examples, of the test questions I might say, do mitochondria have their own DNA? Or where would DNA be found inside a cell, like a animal cell?
Nucleus?
Yeah, and
Nucleoid?
no, we just talked about it. What's that organelle
Mitochondria.
called? Mitochondria, yeah.
Oh,
So there's two places that you guys have DNA in your cells. One is the mitochondria and one is the nucleus. And here's another thing that I should mention is that, again, these are only found in eukaryotes because prokaryotes don't have organelles. But the number of mitochondria can vary between cells. So the cells that use a lot of energy, like your brain cells and your muscle cells, might have hundreds, if not thousands, of mitochondria in them, whereas maybe a skin cell would only have a few because it doesn't need as much energy. So you can the number of mitochondria can vary per cell depending on how much ATP it needs to function. All right, so chloroplasts are only found in plants. This word plast tells you it's a plant thing. Chloroplasts they're kind of like mitochondria. They have an outer membrane and they have an inner membrane. and the space in between is called the intermembrane space. But they also have these things called thylakoids, which are inside here. Which are these things right here. So the thylakoids are these little discs. They're stacked on top of each other. The stacks of these discs are called granum. And again, they have an inner and an outer membrane. I drew it over here because it's kind of hard to see it. But that's what it looks like. And then inside the thylakoid is called the thylakoid space. So it's thylakoid, and then you just add the word space to it. And so we're going to cover this in chapter 10 when we get to photosynthesis in detail. But this is just sort of a foreshadowing. So remember, plants take in carbon dioxide, and they combine that with water, and they produce sugars and give off oxygen. Let me just balance this real quick. So this is photosynthesis, which is exactly the opposite of respiration. So you guys take in oxygen and sugars and give off water and carbon dioxide. So respiration is photosynthesis run in reverse. So it requires CO2, and then that stores energy in organic molecules. We know what things have energy, right? We talked about on your nutritional label, you have sugar and protein and fats. And then the other one we don't really generally talk about is alcohol is another source of energy. And this is why yeast make this as a byproduct, because it's actually an energy source. The chloroplasts are only found in plants and algae. And remember, the algae are the protists that you just covered in lab. Chloroplasts have their own DNA. So again, they think that maybe these are ancient bacteria that had a sharing relationship with a ancient cell. And so it said, okay, well, I'll make your sugars for you if you give me a nice, safe place to live. The stroma is the area inside here. So the stroma is the area in between the intermembrane and the thylakoid membrane. It's kind of like the inner part of the chloroplast. And by the way, the inner part of this, of the mitochondria, the very inside here, is called the matrix, just like the movie. All right. So does anyone have any questions about that? And then plants can have lots of chloroplast in cells. The photosynthetic pigment is chlorophyll, and it's green. So you can imagine that anywhere in a plant that's green is going to have chloroplast, and anywhere that's not green is not going to have chloroplasts. So usually these are found in the leaves, and there could be lots of them, but not in the roots or anything like that. I mean, that wouldn't make sense because you don't have any way to get light there.
So chloroplast is only found in the kingdom of Plantae?
No, they can also be found in algae.
Oh, okay.
Which is in the kingdom Protista. Any other questions? But they're only found in eukaryotes. In fact, none of these organelles that we're talking about are found in prokaryotes, and that's a great test question. Do prokaryotes have organelles? And the answer is no. And do prokaryotes have cell walls? The answer is yes. Do eukaryotes have cell walls? Some do. Plants do. Fungi do. All right. So that covers the organelles. I mean, there's others that we're not going to talk about, like peroxisomes and stuff like that. But that's enough to just to, get your feet wet in what's inside the cell. And so so now we're going to move on to what we call the cytoskeleton. So you guys know that you have a skeleton, and if you didn't have any bones, then you'd just be a blob. You wouldn't be able to move around. So you have an endo, which means inside, right? Skeleton. And then like bugs and crabs and stuff like that, they have an exoskeleton. And you might have seen, people that, are paralyzed or whatever, they're working on creating exoskeletons. So instead of having an internal skeleton, they'd have an external one, like a robotic shell that allows them to walk around and move. And then that also there's a military use for this as well. But that's the difference. And so when we talk about skeleton, we're talking about something that's going to be supportive, right? Either on the inside or the outside. And when we say cyto, well, that word means cell. So what we're talking about when we talk about cytoskeleton is we're talking about the supportive structures in the cell. So the cytoskeleton is made up of fibers, right? And these fibers are usually proteins, which are made of amino acids. And you guys learned that in chapter 5. And they go through the cytoplasm. So it's kind of like a scaffolding. It organizes structures and can also act as to allow movement either within the cell or between cells. It can give support. There's 3 main types. So we have microtubules, intermediate filaments, and microfilaments. And you're just going to have to learn these and what they are. There's a summary slide and there's a summary table in your book that sums all this up, but we're going to go through these one by one so that you know what they are. This cytoskeleton is really important. Imagine if so your cell membrane is about 1/10,000th the thickness of a sheet of notebook paper. So imagine if you didn't have a cytoskeleton and someone gave you a high five. What would happen to all your cells?
You get destroyed.
Yeah, they would explode. And then you lose your hand. Or if you tripped and fell down, you turn into a puddle. So you need this internal network of proteins to hold your cells shape and to allow you to do the things that you do, that living systems do. So different cytoskeleton parts have different functions, but they all together always help support the cell, right? To help maintain its shape and rigidity. So the microfilaments, right? And this can get confusing, so make a note card or whatever you need to do to remember it. Microfilaments are made up of the protein actin. Remember, proteins are made up of amino acid chains that come from genes, and you guys saw that on your first test. They are solid rods, so these proteins kind of snap together like Legos. And think about like if you super glued your Legos together, so it will make solid rods. They're made from, you don't need to know this. This is the kind of actins. Scientists usually labels things like alpha. So there's an alpha actin and a beta actin. And then the next one they use is usually gamma actin. So, this is when we talk about alpha helices and beta sheets and stuff like that. This is a common theme in biology and medicine. So this is gamma actin, but you don't need to know that. Let's just say it's actin. And it's linked into chains, right? Like Lego blocks super glued together. 2 of these actin chains are wound together to form a helix. So I don't know if you can see it on this slide, but it's actin chains are like this. And again, they're made out of proteins. And the reason they're linked together like that is to make them stronger. It's kind of like braided rope. And so you have these actin filaments within, let's just say, a muscle cell. And so these are the orange things here. And then in between there are motor proteins. And remember, everything in biology has a specific shape and it has a specific function. So these motor proteins only fit on actin. They don't fit on other cytoskeleton components. So this particular motor protein is called myosin. And myosin is responsible for pulling. So essentially, think about it like this. So think about it. This is a bunch of people, and they're pulling on this rope, right? And we got other people over here, and they're pulling on this rope this way. So as they pull the rope together, the actin filament, it gets shorter. And so if you guys, all you guys right now, put your arm out and lifted your arm up. So you lift your hand up, your bicep is contracting because those actin filaments are getting pulled together. So they're getting shorter. So your muscles are contracting that way. And now take your hand and lower it down. And now your actin filaments, those motor proteins, are moving the opposite way to relax your muscle cell. And that allows you to move your hand down. And so that's, if you lift and you lower your hand, that's muscle cells inside actin and myosin are moving those muscles and allowing you to, lift objects or put them down. So microfilaments are in charge of support and muscle contraction. They're also important in localized contraction. So one thing is we have so we talked about the zygote, the fertilized egg that you guys all came from. So the zygote is a sperm plus an egg. And so it's a fertilized egg. And from there, your cells have to divide to make 1 trillion cells before you're born. So those cells have to be divided in some way. So what happens is that these microfilaments, this actin and myosin, make a ring around this cell. So imagine if this was Play-Doh, and you took a piece of dental floss and wrapped it around that Play-Doh and then pulled it really tight. What would happen to that Play-Doh?
It would split.
Yeah, it pinches in two. And so these microfilaments pinch cells in two during cell division to allow you to take one cell and make it two, and then these two become 4, and so on and so forth. And so it's this wrapping of this actin filament, and then these motor proteins just walk it together like you would pulling a piece of dental floss or a rope across a piece of Play-Doh, and it would pinch it in two. The other thing is you guys didn't get a chance to see amoebas, and normally when we have in-person class, you can see amoebas. They look like these blobs, and amoebas are kind of like white blood cells. So white blood cells do the same thing. So they kind of ooze around, and the way that they move is that it's called amoeboid movement. But what happens is that their cytoplasm, they can change the shape of it. And so what they'll do is they'll build these actin filaments out into these little, what they call arms. And so they make these things called pseudo. You guys know what pseudo means? It means false or fake. And then podia, do you guys know what that word is? I bet you do. You just hadn't thought about it. What's a podiatrist do?
False feet.
Yeah. So what's a pseudopodia?
So false feet.
Yeah, false feet. So they push this out with using this actin and myosin, and then they can actually eat this way. So let's say a bacteria came in here. We'll make it an orange bacteria. So an orange bacteria comes in here, and these pseudopodia would actually surround this thing and pull it in, and then the amoeba would stick it where? What organelle would you want to put any bacteria that you brought it into? Come on, it's the stomach of the cell. You guys are going to know this for the test, right? The lysosome. So anything you bring, anytime you bring stuff from outside, you don't know what that is. It could be dangerous. So you want to put it in the lysosome. Okay. All right, and so the last thing is cytoplasmic streaming. So cytoplasmic streaming, let me see if I can show you a video of this. Okay. Okay, so hopefully you guys can see this. Essentially, this is just a YouTube video. You can see that these are chloroplasts, and you can see that they're moving around. So plants don't have a circulatory system. They don't have a heart. They don't have blood vessels. They have vasculature that they can move things through. But anyway, this cytoplasmic streaming is actually done by, hang on a second. I'll put us back to the. So the cytoplasmic streaming is actually done by these microfilaments. They sort of project out like little hairs, like these pseudopodia, and then they just sort of beat like oars and move liquids around inside the cell, so they can move nutrients throughout the cell. And that's generally done in plants. So those are the functions of microfilaments. You guys have any questions? So on the test, I might ask you, what are the functions of microfilaments? You know, and it might be multiple choice. Like, you know, you should know all of these, what it's responsible for doing in the cell. You should know what it's made of. So it's made out of actin. And that's pretty much really all you need to know for that. Microtubules are different. So microtubules, just like the name implies, they're tubes. They're made up of a protein called tubulin. So this is a protein. Again, this is made out of amino acids. Every cytoskeleton is going to be used for support. What's unique about microtubules is they can move actual organelles around. They have a motor protein. This motor protein, so I'm just going to kind of explain how this works. So you guys have 2 feet, right? 2 legs. And ATP, one of the reasons that it's used for energy is that we can transfer a phosphate to this motor protein. And when I add a molecule to another molecule, what does it do to that molecule's shape?
Changes it?
Yeah. So by adding phosphates and taking them away, I can change the shape of these motor proteins. Now, it's really no different than what you do when you're walking. So after lecture's over, when you guys try to walk around, see if you can walk without moving your legs or changing the shape of your legs. And the answer is there's no way you can do that. You could not move without changing your shape. So that's why ATP allows these motor proteins to move. Same thing with your muscles. This motor protein is called dynein. You don't need to know this. I'm just telling you this. Remember, it's a different molecule, so it's going to have a different motor protein. And then we talked about this one. This one is called myosin. And you should know that one. But this one you don't need to know. You just need to know it's a motor protein that moves stuff around. It also is responsible for separating chromosomes during cell division. So you guys have, we're going to cover this in chapter 12 and 13 and part of 14. But you have 23 pairs of chromosomes. We already talked about this. So 23 you got from your mother, and 23 you got from your father. And so those are all numbered, and we'll just call this number 1. They're numbered by their length. So they get shorter as the numbers go on, and so on. And so originally your cell would look like this. So you have, let's say this is chromosomes from mom, and we'll say these are chromosomes from dad. And on this chromosome, you might have a gene for eye color. And the one you got from mom might be, or from, sorry, this is dad. The one you got from dad might be blue. And the one you got from mom might be, let's just say, brown. Right? And so that's what your chromosomes look like. And whenever you go through the cell cycle, and we talked a little bit about this, we have G1, and again, we'll cover this in chapter 12. G1, S, G2, and then M, this mitosis phase. So right here, the DNA is replicated. So we can go ahead and just fake replicate this DNA. So you'll make a copy of your dad's, and you'll make a copy of your mom's chromosome. And then these are held together by a centromere. You don't need to know all this. I'm just sort of showing you. So that's why these chromosomes look like X's. They're actually 2 chromosomes that have been copied in S phase. And now we have, instead of having 46 chromosomes, we've copied all of them to make daughter cells. So we have another set, and that means we have 92 total chromosomes. So we have to separate these chromosomes into the new cells, and they have to be separated equally so that only one copy goes from mom and one copy goes from dad into one cell. So this chromosome and this chromosome would need to go into one cell, and then this chromosome and this chromosome would need to go into another cell. And this is no easy feat. And again, we'll talk about this in chapter 12 and 13. But the way that they get moved around is through, so I'm just going to draw a fake chromosome. We'll have a centromere. And these microtubules, they have these centrosomes. Again, we'll cover this in chapter 12. But they build these microtubules out and attach to the chromosomes, and then they pull the chromosomes to opposite sides of the cell. So that's how we separate and make sure that we don't, like, so let's say this is mom's number 1 chromosome. So we get one copy of mom's number 1 chromosome in the daughter cell and one copy of mom's chromosome in the other daughter cell. So these microtubules are really important in separating these chromosomes. And the chromosomes are really long, and so they're not rigid. They're not like pieces of metal. They're more like headphone wires. So imagine if you had 46 pairs of headphones in your gym bag, and you just threw them in there, and then at some point you needed to separate those out, and let's say you wanted to get one headphone out, your favorite ones for your workout. You pull those out, what's going to happen?
They're all tangled.
Yeah, they're all tangled. So there's a magic feat that these chromosomes do in order to make sure that they're not tangled. And basically it's just wrapping them up around those kite spools that we talked about. Remember the histones, the proteins that chromosomes get wrapped around, in order to keep them organized so that we can do this more easily without ripping the chromosomes into shreds. All right. So that you don't need to know all this stuff right now. It's just a little foreshadowing, but I wanted to explain how they work in separating chromosomes during cell division. For right now, this is all you need to know, but you will need to know this other stuff once we get to chapter 12. They also are in charge of cilia. So remember we talked about cilia when I showed you that electron micrograph of the inside of your small intestine. And we talked about the cells that, you remember, they have those little projections on them. And this is your small intestines. And these things beat, and their job is to move your food along. Well, your cells in your lungs also have cilia, and their job is to beat the pollution and junk that you breathe in out of your mouth. So these cilia are really important. They help move food in humans. They help remove large pollution particles and dust and stuff that you get in your lungs. And then we also have flagella, and these are longer ones. So if you just go, I'm just gonna go back. Come on. So this slide, these things called microvilla, these are also cilia. And those are the things that are projecting out that move particles around. There's lots of them. And then we have flagellum. So the best example of flagellum are really long, sort of whip-like structures. And these are found in sperm in humans. All right, so both cilia and flagella are made from microtubules, which are made out of tubulin. So for this, you should know that microtubules are part of the cytoskeleton system. They're hollow tubes that are made of tubulin. They provide support. They help move organelles. They allow for separating chromosomes during cell division, and they are also responsible for the formation of cilia and flagella. All right, so cilia, again, we talked about this. So cilia are used to move things around. Your windpipe has cilia. Your small intestines have cilia to move your food through. These are in large numbers, so we're talking about thousands of these little hair-like projectiles. This is an organism that's beating the cilia to try to get this food inside of it. They're shorter than flagella, so you know, flagella are really long, cilia are real short, like here. It works like an oar. So we have a power stroke and a recovery stroke. Think about this. Like, so let's say you're pretending you're rowing a boat. So you're going to put the oar in the water and then you're going to pull on it. And that's the power stroke, right? And then to make another stroke, you have to take the oar out of the water, put it back in. And when you're taking it out and putting it back in, that's what we call a recovery stroke. Because it doesn't take as much force to do that. So you're going to heave, right? And then ho, heave, ho, and then that makes the boat go. So that's how these cilia actually move, which is different than flagella. So flagella, again, these are used for movement, but usually these are to move things around, like the sperm that we talked about. There's their small number, usually 1 or 2 per cell, you know, and this is just a general, we're talking about general things. So this is common in protists for sure. They're longer than cilia, which we just talked about. The way that these work is that basically it's like a long whip that's attached to a motor that spins. So it kind of works like a boat propeller where it's constantly rotating, and then this thing is going to whip out and rotate as well. So if you could think about you holding a piece of rope and spinning it constantly, that's how the microtubules and flagella work, which is different than sort of the rowing motion of cilia. And then, like we said, there's a tiny motor there that spins around, just like a boat motor, and that creates force to allow it to move in the same direction. This is an electron micrograph of a sperm. What kind of microscope do you think took this? Scanning or transmitting?
Scanning.
Yeah. Because we're not looking at the inside, we're only looking at the surface of it. Good job. Okay, so the last one are intermediate filaments. Intermediate filaments are made out of keratin. Keratin is the same stuff that makes your fingernails hard and your hair strong. Keratin is a protein, just like all the other parts. So remember we talked about we have the microfilaments, and those are made up of actin. And then we have the microtubules, and those are made up of tubulin. And then we have the intermediate filaments, and those are made up of keratin. And all of these are proteins, which are made up of amino acids, which we learned in chapter 5. So this is a more permanent structure. They're not, keratin is more like a long, continuous protein, whereas remember the actin and tubulin are stored more like Lego blocks. So again, its function is to help maintain support, so bearing tension. There's a framework inside the cell on the outside. It's basically like a microscopic fingernail that helps give your cell its support structure. It helps reinforce nerve shape. So in your body, you have these nerve cells. They're made of an axon and these long neurons. And these conduct electrical currents. And then there's gap junctions between neurons. But some of these neurons can be like 6 feet long, you know, however tall you are. They run from your brain all the way down to your big toe, the end of your big toe. And so you can imagine what would happen if your spinal cord got severed. You wouldn't be able to send signals, right? You'd be paralyzed. So you can imagine how it would be super important to, coat these things with like a fingernail substance to help protect them from getting severed. And that's what keratin does, right? So it's protecting these and giving them strength, reinforcing nerve axons. It helps fix organelle position. So it helps make sure the nucleus stays where the nucleus is, right? So there's keratin that is constructed kind of like a scaffolding to allow the nucleus to stay in the center of the cell. That's kind of why it's important to learn how to use a microscope so you can see this stuff for yourself. But in the COVID world, hopefully you get an idea from the lab exercises that we're doing. And then it also forms the nuclear lamina. So remember we talked about the nuclear lamina. I'm just going to go back real quick. So on slide 13, we talked about the nuclear lamina, right? And I told you that this is made out of keratin. So it's a protein filaments that stabilize the nuclear shape, and I kind of drew it around in the inside of the nucleus. So that I might ask you on the test, what cytoskeletal component is the nuclear lamina made out of? And you hopefully will tell me intermediate filaments, or you'll get it wrong. So microfilaments, microtubules, and intermediate filaments make up the cytoskeleton. This is an overview. It's from your textbook. So it sort of breaks it down. So microtubules, remember it's a hollow tube. You don't need to know that it's 13 columns, but you need to know that it's made out of tubulin. For microfilaments, you need to know that this is made of actin. It wouldn't hurt to know that it's made up of 2 strands of actin, but I probably won't ask you that. I will ask you what it's made out of. Intermediate filaments, those are made out of keratin that we just talked about. You don't need to know any of this. I don't care that you know the diameter, even though you should know 7 nanometers is really small. Here's the protein subunits. So tubulin, again, you don't need to know it's made up of alpha and beta tubulin. Actin makes up microfilaments, and then keratin makes up the intermediate filaments. You should know the functions. So we talked about this, maintain shell shape. It's the same across all of these, right? They all do that. This one is for cilia and flagella to mobilize the cell or to move things outside the cell. Move chromosomes during cell division, we talked about, and organelle movement, that's for microtubules. So you should know all that. For microfilaments, you should know that we talked about pseudopodia, so cell movement. We talked about cell division. We didn't talk about a cleavage furrow, but a cleavage furrow is the indentation that would be caused by your dental floss. In this case, that's the microfilaments sort of pinching this in two. So this indentation is called a cleavage furrow. You don't need to know that right now. We'll talk about that in chapter 12. But you should know that it's important in splitting the cells in two. So you don't need to know cleavage furrow, but you should know that it's responsible for dividing the cells. We talked about cytoplasmic streaming. We talked about it's important for muscle contraction. You don't really need to know that. And then for intermediate filaments, so that's the microfilaments. Intermediate filaments, we have anchorage of the nucleus. We talked about that and other organelles. And then it also makes up the nuclear lamina. So you should know all that. And then it might help them just get some note cards and memorize this, because you're going to need to know that for the test. Okay, so to sort of wrap up this chapter 6, we're going to talk a little bit about the cell wall. So we know that the cell wall is made up of cellulose. You learned that in chapter 5, and that's made up of beta glucose linkages, not alpha. And that most plants produce this. It's outside of their cell membrane or plasma membrane. It, like we said, it's made out of cellulose, and we can also have sugars added to it and proteins added to it as well. So polysaccharides and other proteins can be attached to the cell wall. It functions for protection, right? So it's what gives like a tree its strength and rigidity. So it also protects it from outside invaders like viruses and bacteria. You know, it's like a strong armored external wall of like a castle or something. And then it also helps prevent it from taking up extra water, because if the cell got flooded with water, what would happen? It's like blowing up a balloon. If you keep blowing it up, what's eventually going to happen? Are you guys there?
It will rupture.
Yeah, it's going to burst. It'll pop. And what happens if your cell membrane pops? What can you no longer maintain? That fancy word from chapter 1.
Homeostasis.
Yeah, homeostasis, which is important for life. And the cell needs to be able to, cells need to send things to other cells. So they have these little channel in between the cell walls. And this is in plant cells usually, called plasmodesma. These are important because when we're manipulating plants, like let's say we want to make a genetically modified corn or genetically modified papaya. They're almost all genetically modified because there's a disease that kills these, and by adding an external gene to it, it actually protects the papayas. So if you go to almost any store and buy a papaya, it's going to be genetically modified because the virus killed off all the ones that weren't modified. But in order to modify this thing, you have to get your DNA across all of these cells. And so there's a way to exploit this plasmodesma to allow you to introduce external DNA into plants. And this is kind of how it's done. So that's why this is important. It's important for biotechnology, and you know might want to make plants that are resistant to drought. So we would need to exploit the plasmodesma, or maybe plants that produce more food because the population is increasing, and so is the demand for more land that you can grow on. So maybe plants that don't need as much nutrients can live in salty environments, can tolerate freezing and things like that. All right. So the last thing we're going to talk about is the extracellular matrix. So we've talked about the cell. We know that in eukaryotes, we have organelles inside the cell. So we're talking about all the stuff inside the cell. But if you took a cross section of, say, your skin cells or your like a piece of skin, you would see that cells aren't actually touching each other. There actually is a space in between them. And that space is called the extracellular, so outside the cell, matrix. And this is really important. So you can kind of think about cells as like houses on in a standard subdivision, single family houses. They're going to have yards around them, and they're not going to be touching their neighbors like so. And so this extracellular matrix is a way that they can communicate with one another. So we have communication. It also provides cushioning, kind of like the cartilage in between your joints. So this can get compressed if the cells are getting pushed together. And so that provides cushioning for your cells. And it also provides support between cells as well. It does a lot of other things too. There's a big interest in how this works in cancer cells. So let's say that so this is my foot, and let's say I got a cut on my leg right here. So when I get this cut, if I looked at it under the microscope, I would see that my skin is separated. And you don't maybe not pay attention to what's going on here because it's going to scab. It'll bleed and it'll scab. But what's happened is that you have basically separated these cells from their neighbors. So these are all cells surrounding this cut. And in order to fill in this gap, these cells have to go through cell division, and they divide and make new cells. And when they divide and they meet their neighbor, then they stop dividing, right? And this irregular division, because they're not really sure how they're supposed to divide, because they're not touching each other, usually causes a scar, right? So you might end up with a nice scar there, but this is going to be new cells that have grown from your other cells that surrounded that cut. Now, these cells know when they touch their neighbors to stop dividing, but what if they didn't stop dividing? What would happen then? The cells just continuously divided.
A tumor could form?
Yeah, you get a tumor. And so the signals that tell cells to divide and not divide are actually going through the extracellular matrix. So a lot of cancers can occur because of faulty components in this extracellular matrix. So that's really important for communication, especially when we're talking about cancer and tumor development. So I just want to throw that out there because this is a big hot topic in cancer biology. So I just want to break down these things. So proteoglycan, glycose is sweet, protein, this is proteose, so it's a sugar protein. That's what this is. This is just part of the extracellular matrix. Polysaccharides, you guys know what those are. We learned this in chapter 5. Those are long chains of sugar. Collagen, right? A lot of people, want to replace collagen. It's an anti-aging thing. So that's really important for the elasticity of your skin. As you age, your skin gets thinner, and it's because this extracellular matrix gets thinner. So then we have, proteins that are anchoring. This is the phospholipid bilayer here. This is the internal cytoskeleton. This could be microfilaments, or it could be microtubules, or it could be intermediate filaments. But they're going to, give this internal support. And then these are proteins that go across the membrane. So what's the word for across? Like if you're going across the Atlantic Ocean.
Trans.
Yeah. So these are called transmembrane proteins. And their job is to transport things in and out of the cell. So let's say you needed sugars. Well, it would take sugars and pass it through it. And these are unique. So if this was transporting glucose, it could not transport anything else. It couldn't transport iron. It couldn't transport vitamins like vitamin B or C or whatever. It would only transport glucose. So these transmembrane proteins are really important, and their shape is really important. And we're going to learn more about this when we get to chapter 7, next chapter. Yeah. Anyway, so that's what the extracellular matrix looks like. You don't need to know what it's made out of. I could care less that you know this. You don't need to know integrin. You do need to know microfilaments, but not where they're located in the cell. I'm not going to draw this. You do need to know the cell is made up of a phospholipid bilayer for the exam, and that the function of the extracellular matrix is support, cushioning, and communication. That it's mostly made up of glycoproteins, which are proteins with glyco means sweet. So these are sugars attached to proteins. Any questions? All right, that's the last slide for this chapter. So I'm going to go ahead and, since we have a few minutes, I'll go ahead and start the next chapter.
Are you still lecturing?
Yes.
What do you mean, yes?
Okay. Okay, so the next chapter is chapter 7, and this is where we're going to talk about the membrane specifically and its function and structure. So you guys already know that all cells have a phospholipid, which we learned in chapter 5, and it's a bilayer. So it's 2 layers of phospholipids, and that means it has that head and those 2 fatty acid tails, and that makes up the plasma membrane, right? And the reason for that phospholipid bilayer is to separate the inside from the outside, and the reason it's made up of fats is because we're mostly water, and we talked about you don't want to build your house out of paper because when it rains, you wouldn't be able to maintain homeostasis. So the plasma membrane's job is to maintain homeostasis, to select what goes inside the cell and what comes out of the cell. So it separates the living cell from its non-living surroundings. The phospholipid bilayer is super thin, 8 nanometers thick, like we talked about, 10,000 the thickness of a sheet of paper. Its job is to select what goes in and out of the cell. So we call it selectively permeable. That means that, it chooses. So if there's glucose out here, then it needs a channel in order to allow glucose to come in. If there's no channel, then glucose isn't going to go into the cell. And this is, malfunctions in the production of these channels lead to diabetes. Because the glucose doesn't have anywhere to go. It has to stay in the blood, and that causes all kinds of issues. So selectively permeable means some things can go in, other things can't, right? If you don't need any iron, then it's not going to make a channel to allow iron to go into the cell. The one exception to this is that it always has these channels called aqua porins. What's aqua, you think?
Water,
And what's a pore?
an opening.
Yeah, so opening or a hole. So literally these are water holes, and they're always open, so water can go in and out, and you guys just finished up the osmosis thing. So this, because of these aquaporins are always open, it sets up a special thing where water moves across this membrane, and we call it osmosis. And we're going to talk about that in a second. So it has a unique structure which determines its function, and we talked about why it's a phospholipid bilayer and all that other stuff in previous chapters. All right, so we know that phospholipids have a hydrophobic end and a hydrophilic head. Sorry. And so this is going to always go towards water because it's water loving, and this is always going to go away from water. These fatty acid tails, which is that it's water fearing, and this has to do with the fact that these are carbons and hydrogens, and this is non-polar bond. Carbon's 2.5, hydrogen's 2.1, so the difference is 0.4, and that means the electrons are shared relatively equally. That's different from the phosphate because it has that negative charge on it, and water can surround anything with a charge. So that's why this is hydrophilic and this is hydrophobic. Why is that important for the cell? Well, we have to have a bilayer because we want these to line up automatically towards the water, right? The hydrophilic side. And then that gives us this inner part, which is fatty acid tail, which are not water soluble. And so we basically constructed our house out of something that won't dissolve in water. And that's good. And this word amphipathic means you have a dual nature. So you could have a positive charge on one side and a negative on another. We talked about the pH of amino acids. So the amino in the carboxylic acid, this actually donates hydrogen, so it's acidic. And this one pulls out hydrogens, so it's a base. So this would be an amphipathic molecule. So why would this be called amphipathic? What's its dual nature?
That it is hydrophobic and hydrophilic.
Yeah, that's exactly right. So part of it's hydrophobic and part of it's hydrophilic. So this is the word of the day, and I'm going to stop here, and you guys try to use this in a sentence sometime today. All right. Any questions? All right, I'm going to stop here, and then I'm going to jump over to office hours. And then if you guys have any questions you want to ask me in office hours, you can jump over there. If you want to go over your exam things, that we can do that in office hours, so you can see what you missed on the test or whatever. And if not, then I will have office hours on Wednesday, and then we'll pick up lecture on Thursday, same time and place.
Thank you.
All right.
Professor.
Have a good one. Bye.